sometimes they carry their own machinery, sometimes they co-opt the cellular machinery. But at the end of the day, for those of us interested in HIV, and if you look through the literature, you can find thousands and thousands of papers on HIV. And then if you do the double search about HIV and capping, you end up coming up with perhaps 1 or 2 papers over the last 20 years, okay? So it's a surprisingly, the dearth of papers on capping and what is known about HIV capping is essentially a barren desert, okay? And so that presents us with an opportunity then to see what exactly might be going on, okay? In the literature, essentially there is no conclusive evidence on even as fundamental a concept as whether HIV message is capped or is not capped. Keeping in mind that there are large classes of viruses who in fact run around with uncapped RNA. So we came across this particular protein in the course of our interest at looking at RNA binding proteins that could interact, or RNA interfacing proteins that could interact with HIV. And this particular protein is interesting in the sense that its N-terminal domain is a putative transcriptional cofactor because it interacts with P300. But at its C-terminus, it is actually an RNA methyltransferase. So we thought, what would this protein be doing? Would it be a transcriptional factor, as the N-terminus might indicate, or would it be sort of a post-transcriptional factor, as the C-terminus might indicate? So we basically fragmented the expression of the protein into 4 different fragments, the N-terminus, the middle plus the C-terminus, and then the C-terminus. And we asked a simple question, which is that if it is predicted to be a methyltransferase, and if we look more closely, the methyltransferase looks like it's a cap methyltransferase, then if it functions as such, it should be able to bind to M7 caps. Okay? So we generated M7 caps, covalently linked to agarose beads, and we performed pull-down. And the pull-down results then were interesting in the sense that it confirms that every time you have the form of the protein that contains the C-terminus, then that particular protein binds to the N7 cap and is captured in this kind of pull-down assay. If you simply have the N-terminus, then what you find is that the N-terminus completely falls through. So suggesting that at least for this type of analysis, the business end is then at the C-terminus binding to caps. Now, it's one thing for you to bind to cap, it's another thing for you to actually modify the cap. And when you look at cap-modifying enzymes, of course, it comes in at least 2 different flavors. One is the one that methylates at the 7 position, okay, so it's a traditional M7 cap. And then there are proteins that actually hypermethylate the substrate that is first M7 methylated, and then you have 2 additional methylases added here. So this is a trimethylated or a TMG cap. So it turns out that the protein that we're working with is not just a methyltransferase, not a cap methyltransferase, but actually a cap hypermethyltransferase, in the sense that you have to start here, and the protein then takes you from the M7 to the M227, this trimethylated cap, okay? So this is just a simple experiment illustrating that. We've done a lot of other substrate to show you that, in fact, this is the unique substrate, okay? If you just start with a G, it's not going to methylate. But if you start with the methyl 7G, then you get the methyltransferase. And the methyltransferase then is correlated with the presence of the C domain, which, as we predicted, would have the enzymatic catalytic activity. So all of this is making sense, okay? All of this is sort of really fairly basic protein characterization and function. But those of you sitting in the room would say, well, what does this have to do with HIV? So we asked that question too. And so if you want to see what it has to do with HIV, I think minimally you need to say, is HIV RNA capped or not? And is HIV RNA, if it's capped, does it exist in a trimethylated cap form? So to answer that question, since, as I pointed out to you, the literature on this particular topic for HIV transcripts is essentially barren. Okay? So what we did was we said, the biochemistry is not very easy to do, but the molecular biology to look at cap RNA is quite simple to do. Because out there are commercially available, really very specific antibodies that can recognize the M7 cap, and there are very specific antibodies that can distinguish between the M7 and the trimethylated GCAP. So the idea then is that if you take productively infected HIV cells, you do the immunoprecipitation, you are actually going to pull down all RNAs that have M7 cap, which is going to be a lot, and all RNAs that have a trimethylated GCAP, okay, which is also going to be a lot. But by doing sequence-specific PCR, you can identify them as HIV transcripts or not. And indeed, when you do that kind of experiment, okay, it becomes quite clear that HIV transcripts actually come in two different forms. Some of them are methyl 7 GCAP, and some of them are in fact trimethyl GCAP. Okay? So suggesting then there is a level which HIV behaves as all Pol2 transcripts do, which is get, not all, but most Pol2 transcripts do, which is get the methyl 7 GCAP. And then there's an additional step that adds two additional methyl groups to make the TMGCAP. So if that is the case, and if the PIMT protein is doing that event, okay, starting with the methyl GCAP and then adding two more methyl groups to it, two HIV transcripts, then how would that impact HIV replication and expression? So we did a simple experiment, which was to say that if we start with the HIV molecular clone and you transfect into cells, you're going to get very respectable expression of P55 and P24. Now, if we overexpress PIMT in its entire whole length form, what would we in fact see? We would see, in fact, a very robust increase in the expression of HIV, both in looking at P55 and P24. If we then use the deletion mutants which we made, we would find that anytime we muck around with the intact methyltransferase, we in fact remove the enhancing expression ability of PIMT. And in fact, it needs to have the whole protein, suggesting to us that intracellularly, the entire protein is necessary for the methyltransferase activity, and that the methyltransferase activity is responsible for increasing expression. Now, of course, these days, you can only do the overexpression study to a certain extent without also fulfilling the burden of the knockdown study, which is something that we also did, which was to take two siRNAs directed to different sequences within PIMT, demonstrate that we can knock down the endogenous PIMT protein, and then showing that, in fact, that basal P55 and P24 expression in the setting where you knock down the cell endogenous PIMT leads to a reduced expression of HIV protein. So suggesting that in the context of HIV, okay, the trimethylation of the cap mediated by this factor is in fact important to regulate the optimal expression of HIV transcripts. Okay, now those of you would argue to me that in fact we haven't shown the linkage, because anytime you use deletion mutants to prove that you have a loss of function, that is in fact a very crude way and is lacking in requisite sophistication. So of course the much better way would be if we could just simply make 2 point mutations. Okay, so here nothing else has changed except 2 amino acids, which we know would only change the catalytic activity, as we can show you here, of the trimethylation reaction, will not change the localization, will not change any other property of the protein, as a truncation might in fact do. And so this simply changes only the catalytic activity of the hypomethylation event. And if you do that and you use that point mutated protein, then you can in fact demonstrate that it is in fact the singular loss of the ability to transfer the 2 additional methyl groups to the cap that results in the loss of the enhancing ability for expression of HIV transcripts. So I think to that extent then we've established fairly well the linkage between this particular protein, its methyltransferase activity, HIV expression. Okay? But I think the further link of the proof would be to demonstrate that in fact the protein, when overexpressed, does lead specifically to the increase of trimethylated cap HIV transcripts. Okay? And this is in fact what we did in this particular experiment. Okay? So we have a protein that's a methyltransferase. It is a hypercap methyltransferase. Okay? We show that in fact the catalytic domain is important for enhancing HIV gene expression. And then we show that when we overexpress the protein, the HIV transcripts, which normally exist in both forms, either a 7-methyl G or a trimethylated cap form, is highly increased and converted to the TMG form by this particular protein. And our argument is, in fact, that the TMG form of this particular protein is the preferred form for optimal expression of these HIV transcripts. Okay?